So in exam 2, let's go through the logistics. It's going to be the same with exam 1. So in terms of logistics, both exams are going to require respondus lockdown, and webcam. In addition to that, you will need to have to do the environmental scan. And a lot of you did such a great job. And one of you even followed the directions to the T and had a mirror in the back. And I appreciate that so much. Environmental scan. And again, the environmental scan is not meant for me to like invade in your privacy, but it's meant to protect you in case respondus kind of, flags you for potential academic disintegrity. Okay. All right. So any questions on the respondus lockdown, webcam, and environmental scan?
Yeah, I do have a question.
Yeah.
I just recently bought a pair of those noise canceling headsets just because anytime I take a quiz or whatever, the neighbor is using a leaf blower or mowing their lawn or something. So is it
Yeah.
permissible to wear those during the exam?
Yeah, you just have to mention it. Otherwise, respondents will flag you if you have headphones on. So just say that I'm going to wear headphones and they are noise canceling, and then that will not cover you. Yep. Let's see what else. Okay. Any other questions about respondents lockdown, webcam, and environmental scan? So just to remind you, because some of you forgot that you can have a blank piece of paper with you if you like to jot down notes and such when you start the exam. So how that works is that during the environmental scan, you'll just need to show me the front and the back that it's blank. And then afterwards, if you can show me one more time that, it's not a new sheet or anything. So that's for the environmental scan. I think someone put something in the chat, but I don't have that screen up. Can someone read the chat for me? Nope. Okay, maybe I can pull it back up. Let's look at the chat. Oh, it's just Emma's note taker. Okay. All right. So for the lecture exam, you will have 50 minutes.
I did have a question.
Yeah,
I'm so
of course.
sorry. Are we allowed to have water while we're taking our exam?
You can have water, you can have food, whatever you want that helps you.
Okay, because I didn't know if like the water had to be in like a clear glass or if I can drink out of my water bottle or anything specific.
As long, I mean, like, I'm gonna be able to tell if you're like have notes in your water bottle. So, there's a difference between and
Right.
So
Okay.
yeah, have food around you. I mean, that I find that comforting whenever I take exams, so I have nothing against that.
Okay,
Have
thank
food,
you.
have water. Yeah. All right. So the lecture exam you have 50 minutes on, so that it's like equivalent of 1 minute per question. And you'll notice that like 80% of your class did not go anywhere near the full time. So hopefully that helps you kind of get rid of that anxiety. As a matter of fact, a lot of you went through it too fast. So if you're the type that if we met and we talked about you going through it too fast, make sure you're following my strategies of 1, reading it in your head, and then 2, reading it out loud. Okay. Just to kind of slow you down and process the question a little bit more. So you do have 50 minutes like usual, and it has 40 multiple choice questions with 10 fill in the blanks. And they are arranged by chapter. Now, in this exam, there's the skeletal chapter, joints chapter, muscle chapter, and nervous chapter. So muscle actually has 2 chapters and nervous has 3. So I divide the questions evenly per chapter. So that you're going to see a little bit more questions on muscles and a little bit more questions on nervous than you will on joints and skeletal. Okay, because I do divide them equally. So I don't like emphasize on one thing over the other. Okay. So 40 multiple choice, 10 fill in the blanks for lecture. And then for lab, it is 30 questions. Oops, let's go through how much time you have. So for the lab, you do have a little bit more time because it is something that you have to type in. So you have 45 minutes. Okay. And there are 30 questions. And these are all going to be identification. Like your first lab exam had some terms that you had to know, but your second lab exam is all going to be on bones and the nervous. Okay. And no joints. I don't think you had a lab for joints. And then for muscles. Okay. So all identification. And as always, the spelling counts. Okay. Any questions about your lecture or the lab? Okay. So overview of the skeletal system. Make sure you know the types of bones. There are long bones, flat bones, irregular bones, and short bones. And it could be simple definition. For example, which type of bone has the height equal to its width? What would you say? What type of bone where the height is equal to its width?
Long bone. Oh.
Good guess, though.
Oh, short bone.
Yes, that would be short bone. Yep. So if instead I said, what type of bone has it where the height is greater than the width, then you would say
long bones.
exactly. Okay. Then I could also ask the same concept in terms of, okay, the humerus is what type of bone?
Long bone.
Exactly. So those are the two types of questions that you would get in terms of that content. Okay. So one is application, the other one is just pure memorization. Then you should also know the function of bones, that it has movement in association with muscles. It functions in stability or stabilization, in that it forms, your bones form a framework for everything to hang on. Okay. It functions in protection. How? Well, your bones form cages. Like what? What are some examples of cages formed by bones that allow for protection?
Ribs.
Ribs.
What else?
Our skull.
Yeah. Your pelvic.
Our pelvic, right? Our hips.
Yep. Your hips, your ribs. Yep.
Our vertebrae.
Yes, exactly. Okay. It also functions in red blood cell production. How does that occur? Where does red blood cell production occur?
In the bones, right?
Yep. Be more specific.
Bone
the
marrow?
osteons?
Yes, bone marrow.
Yeah.
Okay. And it also functions in mineral storage. How does bone allow for mineral storage?
It allows for calcium, right? And
Yeah.
within the cells, the osteoblasts are the ones
Not within the cells, but within the matrix. Okay.
within the matrix.
So,
Okay.
so
Isn't it-
bone. I'm sorry. Go ahead.
Oh, sorry. Does this associate with the hydroxyapatite?
You got it. Exactly. Yep. So it's within the bone tissue. So bone has the different types of bone tissue, and it uses calcium phosphate or phosphorus. And what it does is that it solidifies it and stores it in the form of hydroxyapatite. Yep. Okay. So that's how it allows for mineral storage, because it's storing the calcium. Okay. And that does mean that, because here's a misconception that people have, that bone is, once bone is made, that's it, right? No, bone is constantly shifting. So they don't realize that bone tissue is constantly releasing calcium and taking up calcium. Okay. And that's based on blood levels. So let's say you go for a week without, intaking any calcium, your body is actually going to break the bone tissue down to release the calcium back into blood. Okay. And then if this happens over a long period of time, your bone is going to be really porous. So it's going to break very easily. Okay. Now, if you have a lot of calcium in your blood, then it's going to go and be stored into bone. Now, if you intake too much calcium, what could potentially happen is that the bone tissue, the calcification or the solidification of the calcium can be too much. So then your bone becomes, too dense. Okay. So there are conditions we call osteoporosis and osteopetrosis, where, you can either have not enough calcium in bone or too much calcium in bone, because it is a dynamic tissue. It does change. Okay. So that's what mineral storage refers to. Then you should also know the anatomy of a long bone. The best way to do this in terms of structure and function is just to draw it out. Like do, 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 and like so. Okay. So we just label, what would this be called?
The articular cartilage.
No, not quite. So I'll draw the articular cartilage.
Epiphysis?
Yes. You were on the right track because it is found here, but this is what the articular cartilage would look like. So surrounding all of the ends of a long bone, that's where you'll have the articular cartilage. But what I was trying to get in terms of A is just, what is this, the ends of a long bone called? And that would be the epiphysis. Okay, good. And then the shaft of a long bone then would be what? Oops. I should write that.
Diaphysis.
Yes. So that would be the diaphysis. Perfect. Okay. Then if I were to draw something like this, draw a little hollow space here and call that letter D. What would letter D be?
No.
Medullary cavity.
Excellent. You got it. It's the medullary cavity, and that's where the bone marrow would be found. Now, bone marrow is not always there, and that's how you can tell the age of the person. So when you're young, your medullary cavity is going to be filled with bone marrow that has red blood cells. Okay. Now, as you age, when you get older, like in your 40s and 50s, it starts to hold more lipids than it does, the red bone marrow. So it starts to hold more of the yellow bone marrow than the red bone marrow. And then as you get really older into your senior years, then it just becomes gelatinous bone marrow tissue. Okay. So that's the medullary cavity. So the medullary cavity contains anything ranging from red bone marrow to yellow bone marrow to gelatinous bone marrow. Okay. Depending on your age. All right. How about this thing right here? In between the epiphysis and the diaphysis, you have this structure. A, B, C, D, E. What would you call this structure?
That's the one you only find in teenagers, right?
Not only in teenagers, but anyone teenager and younger.
Okay. I can't remember the name.
Metaphysis.
Excellent. It's called the metaphysis, or it's also known as what?
Epiphyseal plate.
You're on a run. Exactly. So it's either the metaphysis or the epiphyseal plates. They can go by either term.
Should we know both terms on the
Yes,
exam
ma'am.
to be safe?
Yep.
Okay.
Absolutely. The layman's term for that is called the growth plate, which is why one of your colleagues has said that, is that the one that you find in teenagers? Absolutely. That's your growth plate. So what's unique about the epiphyseal plate or the metaphysis is that it doesn't contain bone tissue, because it's going to eventually allow for the lengthening, the growing of kids. So what does it contain? What tissue type would you find in the epiphyseal plate?
Hyaline cartilage?
Excellent. Yep. Hyaline cartilage. In an adult, that area is going to transition into bone. All you see in there is bone. There's no more hyaline cartilage in there because they've grown. They're not going to elongate any longer. Okay, so that's
So
the
Dr. Boo, that's pretty common for any type of bone that in the beginning is connected with hyaline cartilage, right? Like the
That's
ones
right.
with your ribs. So
That's
it's a
right.
general thing? Okay.
Yes. Yep.
Okay.
Okay. And then the red we talked about, but let's talk a little bit more. So what's letter B? I know you know because we named it already. What's letter B?
The articular cartilage?
Exactly. So it's the articular cartilage, and you'll find them at the ends of a long bone. So at the covering the epiphysis. What's their purpose? What's their function?
Reduce friction.
Yes. Yep. Because oftentimes bones articulate with each other. In other words, they move along each other. So when they articulate each other, they rub against each other. And if it's the actual bone tissue that's rubbing against the bone tissue, that's going to be very painful. And that's why around the ends, you'll find the articular cartilage. It's called articular because that's where two bones articulate with each other. And it's cartilage because it's surrounding it to kind of protect it, preventing the friction that can occur. Now, in some people, as they get older, what happens to that articular cartilage?
It starts to disintegrate. I don't really know the right term.
That works. It wears down.
Wears down.
Over time, it wears down. And what condition is that?
Arthritis?
Yes, it is. Absolutely. It's called arthritis. Very good. Okay. All right. So articular cartilage, function and structure. Okay. How about this one? What do I mean by this one?
So I'm assuming you're trying to draw the periosteum?
Yes, that is correct. So what letter are we? A, B, C, D, E, F. So this will be F. So F is the periosteum. Peri meaning around, osteum meaning bone. So the periosteum is connective tissue that wraps around the diaphysis of a long bone. Okay. What is its function?
So the inside part has the tissue forming cells, right?
Yep. So I haven't drawn that yet, but I will right here.
So you have two layers, the inside and the outside. So one
Yep.
is for the cells, and the other one is for structure.
What do you mean by
strength?
structure?
Kind of just giving more of a strength to the bone.
Not so much as strength, but it is a place of attachment. So
Okay.
what is always attached to bone?
Ligaments,
Yes.
muscles.
Ligaments and tendons.
Ligaments.
Okay.
Okay.
So attached to bones would be ligaments and tendons. So the periosteum allows for attachment of the ligaments and tendons. So it's either going to connect it to another bone or it's going to connect it to a muscle. Most oftentimes it's a muscle. Okay. All right. Now you mentioned it, the green layer, the inside layer, we'll call that letter G. What is that one called?
So you're referring to the inside now?
Yep.
Endosteum.
Yeah.
Perfect. Yes. Endosteum. Okay, so the endosteum. So the endosteum, you can call it either way, endosteum or endosteum. Okay, so it's the inner layer, hence the word endo because it's on the inside, osteum because it's still going around the bone, right? The endosteum is very specialized because it has very specialized cells that line the endosteum. Do you know what that cell is called?
Sorry, could you repeat that again?
Yeah. Okay. So the inner layer is called the endosteum, and it has specialized cells that line the endosteum. What is that cell called?
Osteoblasts?
Oh, you're so close.
And just osteocytes?
So close.
Oh man.
You only have 2 more. Keep going.
Osteoclasts?
1 more.
Osteogenics?
Absolutely. And you know what? You're gonna get this question right on the exam because you're like, nope, it's not that. It's not that. It's this one. Okay. So it's the osteogenic cells. Now the osteogenic cells can be found in 2 areas, and the endosteum is one of them. The osteogenic cells is the cells that can divide. In other words, it's mitotic. I mean, it can divide and then create more cells. Okay. So the osteogenic cells are capable of dividing. Why is that important? Why do you need that?
Because the osteogenic cells will give you the osteocytes.
Yes, ma'am. So the osteogenic cells will divide and become osteoblast, and then the osteoblast will become osteocytes. Now, why that's important is because let's say you needed some calcium, right? And the calcium has to be pulled out of your bone. So then that means that you need new cells to occur so that they can pull the calcium back into the bone and form bone tissue again. That's one. Secondly, what happens if you break a bone as an adult? If you break a bone as an adult, it actually is going to heal from the outside in because the osteogenic cells are found on the outside, even though it's an inner layer of the overall surrounding connective tissue, it still occurs on the outside. Does that make sense? So if I were to fracture a bone, so if I were to break it like this, okay, that bone is going to heal from this point right here, and it's going to heal inward. From this point, it's going to heal inward. Okay. All right.
You said like in adults, would it be different in children?
In forming fetuses. Okay, so in forming fetuses, they go through what's called the endo ossification. In the endo-ossification process, bone actually is formed on the inside first and then out. Okay.
Okay, thank you.
Yep. So that's the difference. Is that endo-ossification, okay, is a one form of bone formation during fetal development, where it first lays down a cartilage template and then that's replaced by a bone tissue. And through that process, it's going to build from inward outward. So that's how bone is modeled and made, okay, in the fetus. But as you become an adult, or as once you're born, the bone is always going to repair itself from the outside inwards. Okay. All right.
A question,
yeah.
Dr. Ruby, before you go on.
Yeah, go ahead.
So the osteogenic cells are in the endosteum and in the periosteum, and then you have the osteocytes are in the matrix. They become trapped in the matrix.
Wait, 1 second. So the osteogenic cell, no, that's okay. The osteogenic cells are found only in the endosteum, not in the periosteum.
Okay, because I'm seeing here in the notes from the slides, it says the inner layer of the periosteum as well.
Yep. So the inner layer. Yep, that is correct. So the inner layer of the periosteum is the endosteum.
Ah, well, it's all making sense now. Okay.
Yeah.
Oh, I was confused about that too. That makes sense.
Yep.
Okay. So that's where you find those. The osteocytes then become trapped in the matrix. Where are the osteoblasts?
So the osteoblasts are formed from the osteogenic cells. So wherever the osteocytes are, that was once an osteoblast.
So they are in the matrix per se then. So they start in the endosteum and then they move into the matrix as osteoblasts and they mature into osteocytes there.
Yes, you got it.
Perfect. Thank you.
Yep. Okay. All right. And let's see what else. So we got the okay. I think we got the bulk of the structure and function of a long bone. Okay. Now, if I take a cut of that long bone, okay, what I would expect to see is this. So I'm going to try to keep the coloring the same. So purple here is the same thing as purple here. So what are you seeing here? Tell me what this is. And tell me what this is. Since I just cut the long bone in half.
So I'm assuming you're drawing an ostium, and the middle is a central canal?
No, ma'am. I just cut the
No?
long bone in half. So this
Okay.
purple here is this purple here. This purple here is this purple here.
The lamellae?
No, this is still the long bone. So the purpose that I was trying to do is that I was trying to say that this right here is letter D. So that's the medullary cavity. This right here is letter C. So that's the diaphysis. Okay. Now, along between the medullary cavity to the outside of the diaphysis, it's going to be filled with smaller tubes. And I'm going to make the smaller tubes green. So essentially, the bone tissue that forms the diaphysis of a long bone is compact bone tissue. It's made up of all of these tubes. Okay. And if you wanted a 3D rendition of this, it would look something like this. So here's the long bone, here's the diaphysis, and if I cut it in half, you're going to see that it's like a tube that contains smaller tubes on the inside. Okay, so let me ask you something. So this is compact bone tissue that forms the diaphysis, where spongy bone tissue forms the epiphysis. Okay, if I have one stick right here, can you snap the stick in half?
Yes.
Yes. If I had hundreds of these same sticks, now can you snap them in half?
It will be really hard.
It'd be harder.
It'd be a lot harder, right? And that's the premises of the structure of how your long bone is formed. So it has, so the compact tissue along the diaphysis, okay, even though it has single osteons, there's hundreds of them that line the diaphysis and forms the compact bone tissue. It's very heavy and very dense, but it's also very strong. Just like we said, it's hard, it's easy to snap one stick, but if you have hundreds of sticks, it's very difficult to snap. And that's why bone is so sturdy. It's because the structure of how it's made. Okay? So that's why each of these small green tubes right here is what? I'm going to call it number 1. You can do this. Come on. What's each of those small green tubes called?
Aren't those osteons? Oh,
Yes,
sorry.
that is it. They are the osteons. Okay. So I'm just going to pull one out and zoom in on one. Let me here now. Now you're finally looking at an osteon.
Oh, wow, it all makes sense.
That's why you know you have a teacher, right? Okay. All right. So that's an osteon. Now, the osteon also has a center, and that's why I'm being so deliberate of telling you that this is a long bone, where now you're getting to the osteon because they look exactly the same. Okay. So now this is an osteon. Okay. That formed the diaphysis. Okay, so now we're at the osteon level. So now can you tell me what this is? What's number 2?
Central canal.
You got it. That's the central canal. Okay, then you will also find these pockets right here. What are these pockets called?
Is that
Are
the
you referring to
lacunae?
lacunae?
Yes, you got it. Thank you. These are the lacunae. And it helps me to remember because lacunae sounds like lake. So they look like little lakes. What's the function of a lacunae?
It
Isn't that where
contains
the
the osteocytes.
osteocytes?
Yes,
Oh,
that's where the osteocytes are going to be housed. Okay, but let's number that number 3, and let's label that number 3 right here. Okay, so that's where you'll find, you know, it starts out as an osteoblast, and then the osteoblast, it's called the bone building cell because it's going to pull the calcium, the phosphate, it's going to pull the fibers all around it. It's kind of like, imagine that it's building like a castle wall around itself. But what happens when you build the castle wall around yourself is that you become trapped. And when it becomes trapped, it can no longer build anymore. And that's when it is now called what?
Osteocyte.
You got it. Okay. All right. So connecting the lacunas, you have these networks, these tunnels, a network of them because they connect to all of them. They connect to each other. They connect to all around the areas. What are these little tunnels called? Little channels called?
The canaliculi.
Yes, you got it. And what's their purpose?
They connect the osteocytes.
communication.
Yes. So it's not necessary that they connect the osteocytes, but they connect the lacunas, structure to structure, not structure to cells. Okay. So the canaliculi connect to the lacuna so that anything that happens, first of all, you need to have nutrients go to the cells to keep them alive. And then you have to have a way to remove waste, and you have to have space so that their dendrites can talk to each other. They're not literally talking to each other, but like through chemicals. Okay. So you'll have the osteocytes being housed in the lacuna, and then they'll need nutrients. So nutrients come through the form of the central canal, okay, and the perforating canals. And so that brings them to the lacunas through the canaliculi. And then that's also a way of the cells to get rid of the waste products. And that's how things circulate through bone. Okay.
Is the canaliculi, is it like the dendrites from the osteocytes?
No,
It's
ma'am.
not.
Two different things.
Two different
So,
things. Okay.
let me clarify though. So the dendrites are extensions off of the cell, where the canaliculi are actual structures. Okay. So think of it this way. Minneapolis is an osteon. Okay. So, oh, sorry. Minnesota is an osteon. In Minnesota, you have the cities, right? You have Minneapolis, you have St. Paul, and then you have all the highways that connect. And there's multiple highways that you can go on, right? So the Minnesota is like an osteon, where the cities in each of those in Minneapolis, in Minnesota, the cities in Minnesota is like the lacunas. And then what connects the lacunas together is the canaliculi, the highways. So canaliculi are exactly like highways. It's just a way for things to move, nutrients to move to the cities, and waste to move away from the cities to be, exported out. Does that help?
Yes.
Okay.
Yes.
All right. Then, so that's that's the canaliculi. So that's going to be number 4. So we're going to call it number 4. Okay. All right. So that's the structure, the infrastructure of an osteon. Okay. So that's the infrastructure of the osteon. Now, how many of you have heard of a Twinkie?
Yes.
Yes.
Okay. Do you know how Twinkies are made?
No.
So the rolling Twinkie, is it the roll, the Twinkie rolling thing, the Debbie rolls? So you have like layers of cream on the inside, right? So the osteon is like a Debbie roll up or like a Twinkie, because it's going to have layers.
So the layers are like the concentric rings.
They are the concentric rings. Okay. So these are the concentric rings, okay, that like form the bedrock of Minneapolis or Minnesota. Of Minnesota. Like it filled in the spaces. Okay, like it's like the farmlands. It's like the anything that like fills in the space. Okay, so what is that called? Concentric rings of
Lamele.
yes. Okay.
All those terms sound Hawaiian to me.
I know. I think
That's what I
Hawaiian. I agree with you. Actually, it's Italian. Cannaleculi sounds like that one dessert that I love so much. What is that called? It's like a
Cannoli?
cannolis. Yes. Yes. So I relate everything to food, if you haven't noticed. Okay. So I think it was an Italian group of scientists that, named all this stuff. But anyways, so the lamella is just like the rolls of the Debbie desserts, right? So instead of the cream, it's going to be filled with the matrix. So the concentric rings of lamella, instead of being filled with cream, it's filled with matrix. And we know that matrix composed of two parts, organic and inorganic. Okay. And then, so that's the ground substance. And then you have the fibers, right? So what kind of things would you find in the lamella? Let's just list them off.
You'll find the lucune.
In terms of substance, in terms of matrix.
Oh, okay.
Collagen?
Yeah. Collagen fibers. Keep going.
Hydroxyapatite.
Yes. And can you tell me what that is?
They're like crystallized salts,
Dang it, my pen
which is like made of calcium and phosphate.
Yes. Sorry, I have to write with my mouse now because my pen just ran out, just died. So we will try to do this.
Which the inorganic portion helps with the strength to the tissue.
Yep. Nope. It's hard to write with a mouse. Let me see if I can go back to my pen. Yay! Okay, calcium, phosphate. Yep, excellent. So that's what you find in the lamella. The collagen, the calcium, the phosphate. Okay. All right, so that's the microscopic anatomy of a long bone. Okay, make sure you know the cells. There are 4: osteogenic, osteoblast, osteocyte, and osteoclast. Osteoblast is the bone building. B for blast is B for building. And osteocytes come from osteoblasts. And then osteoblast comes from osteogenic. Now, osteoclast right here is a class of their own. Okay. So they come from a different type of lineage. But these 3 right here are connected to each other. So osteogenic cells become the osteoblasts, and the osteoblasts then become osteocytes. All right, let me ask you a question. This is a not anatomy, but physiology question. Okay, so you go hiking. All right, when you go hiking and you get stranded and there's not enough calcium in your diet, which of these cells will be stimulated?
Is it the osteoclast?
Why?
It creates calcium.
Doesn't create calcium.
Oh, go ahead.
It pulls it out, no, from the bone?
You got it. Absolutely. So the osteoblasts would be the ones that are stimulated because it's going to break down the bone tissue. And when you break down the bone tissue, in particular the lamella, you're going to release what back into blood?
Calcium.
Yes. And you need calcium for muscle contraction. You need calcium for lots of different functions in the body. Okay. So if I were to say the reverse and I say, okay, your kid just drank 5 gallons of milk. too much calcium in his blood, what cell would be stimulated?
The osteoblast,
Perfect. Why?
because it helps by removing the calcium from the bloodstream.
And putting it where?
Back
In the bone?
in the bone matrix.
Into bone and forming the bone. Exactly. Perfect. Okay. All right. So make sure you know about bone tissue, that there's compact and spongy. Do a kind of a compare and contrast between those two so that you truly understand why is it compact? Why is it spongy? Okay. And then bone formation, you don't need to know step by step of what happens in the bone formation. I just want you to be able to define what they are overall. Okay, so be able to tell me what is endochondral ossification, what is intramembranous ossification. You will also need to tell me what materials they start with. Okay, so and that's part of their definition. So what materials do endochondral ossification start with and end up with?
So the endochondrial develops from a pre-existing cartilage.
Yes, it goes from cartilage to bone, where intramembranous goes from where to where?
It goes from a membrane,
What membrane?
intermembrane.
What's it called?
Membrane. Mesenchyme?
Mm-hmm. Mesenchyme, very good.
Is that how you mesenchyme? I'm sorry.
Yep. No, you can pronounce it any way you want. Because here's the thing. So when I took my classes, I had a professor that was from Minnesota. And I had a professor from the South. One of them said respiratory, and the other one said respiratory. So that, and then I've had international conferences that I go to, and they pronounce it differently too. So as long as you know what it is and can spell it, you're fine. Because people are going to say whatever way they're going to anyways. Okay. So mesenchyme. There are some things though that I am very particular about the pronunciation too, but overall I'm not. Okay. So mesenchyme to bone. Okay. So here is a cognitive map that I kind of use to lecture for my class. So you can feel free to use that. And I purposely do not label things so that you can use this to quiz yourself. Like what is A, what is B, what is E, how it's connected, and so on. So this is essentially what we've already gone through. Okay. All right. I'm not going to spend too much time on joints because, honestly, it's a lot of memorization. So this would be a nice one to make cognitive maps like I have here, or to make flashcards, just tons and tons of flashcards. What is this? What is this? What is this? Now, the joints, though, it's really important to make a cognitive map. Do your flashcards to make sure that you're being able to recall it. But it's really important to do this cognitive map. And the reason for that is everything looks the same. Synarthrosis, syndesmosis, synchondrosis, So there's a lot of words that sound and look the same. And that's why when you see them all on one page, organized, into where they fit, it helps you to remember. Okay, so I would highly recommend that you complete this form out. Okay, so then when you see the term synarthrosis, that it's not a type of joint, but it's a type of movement, of joint movement. Okay, so you have synarthrosis, amphiarthrosis, and diarthrosis because joints make you sad. SAD. Okay, and that refers to the movement only, not the type of joint. Okay, so if I were to say, okay, what's the joint that you would find in your elbow? What's the joint that you would find in your elbow? What would you say?
Cartilaginous?
Joint type. Yep.
Would that be the synovial joints?
It is the type, but I want to know, that's the structure. What is it made up of? But I want to know the actual name of that joint.
Oh, I don't. Diarthrosis?
See, you just gave me the movement. You just gave me this one right here when I want this one.
Okay.
So what is this?
Hinge?
Yes. Okay. So you may know what you're talking about, but if you don't read my question carefully and know what terms belong to what category, it's gonna be wrong. Okay. So before, when you said that this is a diarthrosis, it is correct, but that's if I were to ask what kind of movement it has. Okay. But I didn't ask for the movement. Okay. And if you said synovial, that is correct, but it's not the movement, it's more of the structure. Okay. So the correct answer to the question that I was asking is hinge. You see what I'm saying? So make this chart, fill it out so that you can clearly see the terms, what category they fit into. And if there's similar terms, you can see the difference between them. Okay?
So you said the correct answer would be hinge?
Yes. Yes, if I'm asking for the type of joint, it would be hinge. If I were to ask what substance what structure, what substance make that joint, then you would say synovial. If I were to say what movement is that joint, you would say diarthrosis.
Oh my gosh. Okay, so I need to be careful.
Yes.
Okay.
That's exactly it. Yep, that was the point I was trying to make.
Thank you.
Okay. Make sure you know synovial joints a little bit more detail. There's meniscus, there's bursa, there's synovial, there's capsule, synovial cavity, articular cartilage. This image right here is really nice at differentiating what is what. Okay. I'm running very low on time, so I'm gonna go straight to muscles. So hopefully that's okay. So on muscles, this is probably one of the most difficult. I find it the most difficult, you know. So there are 2 supplemental videos that I made for this. So make sure you watch them because I do go through muscle metabolism as well as sliding filament theory. Okay. So if we don't get a chance to go through all of it today, you'll have those videos that I created. And you can find those videos under announcements. Okay. All right. So we're just gonna go right into it. Make sure you know function, structure, neuromuscular junction, sliding filament theory, as well as metabolism. These are the 4 main content topics in muscles. Okay. We'll start with the first one because you really can't do sliding filament theory unless you know this foundation information. We just went through a long bone, and you know that the periosteum provides an attachment site for ligaments and tendons. So here's an example of how a muscle is attached to a long bone through a tendon. Now that muscle, okay, when you like flex right here, this muscle right here, this whole muscle group right here, okay, that's what we call a muscle group overall. Okay. Now if I were to break that muscle group down, that muscle group is made up of fascicles. And if I break those fascicles down, it is made up of muscle fibers, or more accurately, muscle cells. This is very confusing. I don't know whoever just named this did us bad, did us wrong. Because here, the term muscle fiber is the same thing as saying a muscle cell. And you're just like, what? We just learned that fibers are collagen, elastic, and fibrocartilage, right? Yes, but those fibers look thin and long, right? So it turns out that this cell that is the muscle cell looks just like a fiber. So instead of calling it a muscle cell, they decide to call it a muscle fiber. Then you're like, what? What are they going to call the fibers in a muscle then? Well, they came up with myofilaments and myofibrils. Okay, so myofilaments and myofibrils is now the new term for muscles, muscle fibers. And a muscle cell is called a muscle fiber. Okay. And like, as if that wasn't bad enough, they actually called all the organelles by a different name. Like instead of calling it the plasma membrane, like you learned, it's now called
Is this called sarcoplasm?
close.
Sarcolemma.
Sarcolemma. So instead of calling it the plasma membrane, it's now called the sarcolemma. Instead of calling things the smooth endoplasmic reticulum, it is now called
Endoplasmic reticulum, something like that? No.
Nope. So instead of calling it the smooth endoplasmic reticulum, it is now called the sarcoplasmic reticulum. Okay. Instead of calling it the cytoplasm, it is now called someone said it earlier.
Is that the sacroplasm?
Yes.
Okay, I got it wrong.
Okay. Right? So it's like, more terms you have to memorize that mean the same thing. It's like, why didn't they just leave it as the cytoplasm? Why didn't they just leave it as the plasma membrane? I don't know. I don't make the rules. I just have to follow them, and so do you. Okay? So now you know. Sarcolemma is the plasma membrane of a muscle fiber. Muscle fiber is a muscle cell. Fibers in a muscle is now myofilaments or myofibrils, specifically actin, myosin. Okay? So it's complicated, and that's why I would highly recommend that you spend, set aside enough time to do the muscles. Okay? All right. So you have the muscle group. Okay. So think of it like, if you're vegetarian, I apologize. Okay. So I don't mean to be carnivorous centric. But if you've ever seen a drumstick, and tore the drumstick apart, you'll notice that it doesn't go into chunks. When you tear the drumstick apart, what does it tear into? Is everyone vegetarian?
Like pieces?
Yeah, like strips, right?
Like strips, yeah.
Yeah, exactly. So those strips are called fascicles. Okay. Then the fascicle is made up of smaller tubes on the inside. And now, if you tore the fascicles apart, you'll actually reveal the muscle fiber, aka the muscle cell. Okay.
So
All right.
what are the myofilaments? Where did they come in?
Yeah. So inside the muscle fiber, aka muscle cell, it's divided by sarcomeres, and then within the sarcomeres, you'll have the myofibrils. And the myofibrils are the actin and the myosin. Those are the fibers that you find in the muscle cell. Yeah, right? And then, so you have the thin fibers and you have the thick fibers. The thick fibers are called myosin, and they have these little oars on them called the myosin heads. And then the actin, the thin filaments, have two additional proteins on them. One that's a smaller strand that wraps around it, okay, called the troponin. Called the tropomyosin. And then it has another protein on it called the troponin. Okay. So actin is always associated with troponin and tropomyosin. Okay. All right. Okay. Let that sink in. Just take a 10 seconds to let it sink in. Okay. So now, if you were ever to see the chicken drumstick, there's this crunchy stuff on the outside of the meat. What is that crunchy stuff made up of?
So you're talking about the skin?
Yeah, exactly. Okay. So it's got lipids, right? It's got some fat and some connective tissue on the outside. Okay, we call those fascia. Okay. Our muscles also have fascia that surround it and bundle it together. Okay. So each level has a different type of fascia. So the muscle group, so when you flex your arm right here, this muscle group right here has a fascia around it called the epimysium. when you peel it off, and then you peel the strip of muscle apart, you have the fascicles, right? The fascicles also have a fat connective tissue that surrounds that, and that's called the paramycium. If I peel the paramycium apart, I'm going to reveal the muscle cells or the muscle fibers, and that's going to be surrounded by a fascia as well called the endomycium. Okay, so you've got the muscle, and then you have the fascia. Okay, the muscle starts with the muscle group, which is surrounded by the fascia called the epimysium. I break that down further. I have the fascicles, and the fascicles are surrounded by the paramycium. I break that down even further. Then I have the muscle fiber, which is just another term for muscle cell, and that's surrounded by the endomysium. Okay, so that's the structure of the muscle and the fascia. Okay, now I'm going to zoom in on just one muscle fiber, in other words, one muscle cell. So what you're seeing here is what I zoomed in here. Okay. So that muscle fiber, aka muscle cell, has a plasma membrane called the sarcolemma. It has this structure right here. What is this thing called? Same name, thankfully.
Mitochondria?
Yes. Muscle cells or muscle fibers are abundant in mitochondria. Why do you think that might be?
Because they need a lot of energy.
That's right. That's right. Now, if it's going to have a lot of mitochondria, it must also have a lot of these things. What are these things called? Chains of sugars called what? Chains of glucose.
ATP.
Nope. not yet.
Glycogen?
Glycogen. That's the best term. Is glycogen, which is chains of glucose. Okay. Now, in addition to that, you will find this in muscle cells or muscle fibers. These proteins that carry oxygen. These proteins that carry oxygen are called what?
Myoglobin?
Exactly. You got it. So in blood, we have the same protein that carries oxygen called hemoglobin. But in muscles, it's called myoglobin because the term myo and sarco means muscle. That's why everything here is sarco and everything is myo. Because sarco and myo are the prefixes that refers to muscle. Okay? So the protein that's going to carry the oxygen is called myoglobin. And you know, between those three parts, the mitochondria, the glycogen, and the myoglobin, it's going to be able to produce a lot of ATP. Because remember, in order to produce energy, you have to have lots of what element?
Oxygen?
Yes, ma'am. That's right. Okay. All right. You also need this right here, this organelle. So muscle cells will always have lots of these organelles. What are these organelles called that contain calcium?
The smooth endoplasmic reticulum, but it's not called that. It's called something else.
You got it. You absolutely got it. You caught it before I could even say it. That's why I laughed a little bit. Okay. It is the smooth ER. Okay, but here it's called the SR, sarcoplasmic reticulum. Okay, so the smooth ER is called the SR, which is the sarcoplasmic reticulum. And the reason why it's important is because it has calcium. So that means that in your head, I want you to store, why do you need calcium in muscle cells? And actually, from your head, tell me, why do you need calcium in muscle cells?
Because those are the ones that go to the toponym and those are attached to the myosin. So you can't move the myosin without the calcium attached to it, right?
Yes, you give the rest of the lecture. I'm gonna take a break. You got it. No, you hit it right on. Very simplistic explanation, but 100% correct. Okay, so that's exactly it. You need the calcium because it has to bind to the troponin tropomyosin to shift away from actin so that myosin heads can grab onto actin and contract. Okay, and pull inward. All right. Okay. So I think those are the main things that. Oh, I almost forgot. You also need to have. You see the sarcolemma right here? How it fold? There's an inward folding right here.
Sorry, I don't mean to interrupt.
Go ahead.
What was that question you just asked again? Why do you need calcium in
Muscle cells.
muscle cells? Okay.
Yeah.
Thank you.
Yep. 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 So what is this next structure right here? What is it called? Starts with a J, ends in folds. So your textbook refers to them as junctional folds. If you are a little bit more old school like I am, I learned it as the motor end plate. Okay, so this is called the motor end plate, or your textbook refers to as junctional folds. That's where you'll find receptors for what chemical?
ACH.
You got it. That's right. Okay. Now, along the sarcolemma, it's going to dip inwards. Okay, and it's going to dip inwards occasionally throughout the whole muscle cell. So these indentations right here, these are called what?
Tubulars.
Transverse tubules. Okay, so transverse tubules for long, T tubules for short. Okay, all right. What's the purpose of the transverse or the T tubules?
That's where the action potential travels to the SR.
You got it. That's right. Perfect. Okay. All right. I feel like you have a strong understanding of the muscle cell. I'm going to move forward, and then I'm going to add another layer to that muscle cell. So far you've gone through the components of the muscle cell, and now we're going to look at the divisions of the muscle cell, how it's divided. Okay. So same muscle cell, I just drew it down below. Now we're going to add another layer of information. Okay. So you got the organelles, you got the overall structure of a muscle cell. Now the muscle cell or the muscle fiber is divided by these zigzags right here. What do we call those zigzags?
Z-discs.
Yes. Okay. So they're divided by the Z disc. And between the space of two Z discs, what do we call that?
Sarcolemma?
Nope, you're so close.
Sarcomere?
That's it. It's a
Sarcomere.
sarcomere. This term is important because when we refer to muscle cells contracting, shortening, we're really talking about the space between a sarcomere that shortens. And that's why we refer to the sarcomere as the functional unit of a muscle cell. So functional unit of a muscle cell. All right, so the Z disc, between two Z discs, you have a sarcomere. And when we say that the muscle cell contracts, it's really contracting in between those spaces. So one segment gets shorter, the other segments get shorter, then this, all the sarcomeres get shorter. And that's why overall the whole muscle gets shorter. Okay. Now attached to the Z disc, you have what myofibril?
Is that actin?
Yes, ma'am. So it's a thin filament. Then attached to the thin filament or the actin, you have what?
Myosin?
Nope.
The elastic filament?
You could, but I'm looking for two different ones, two different proteins.
Oh, are they troponin and tropomyosin?
That's right. Yep. Okay. So as you're sitting there listening to me, are you like this? Are you all contracted like this?
Yes.
No,
No, I'm just
you're
kidding.
not. You're relaxed. You're chilling because this is easy stuff. Okay. So the default status of a muscle is to be relaxed. Okay. So the only time it should contract is when your brain sends signals to your muscles to contract. So in order to have a default of relaxation, you have to prevent contraction. And that is what tropomyosin and troponin does. It prevents contraction by covering the binding sites on actin. Okay. So the analogy that I give is like troponin and tropomyosin are like the parents. Okay. And actin and myosin are like star-crossed lovers, Romeo and Juliet. Okay. So actin is Juliet, and Juliet's parents, troponin and tropomyosin, don't want myosin to hook up with her. So they keep her covered, right? And so myosin wants to hook up to actin, but can't because the parents are there. So what can happen is that you provide the parents with like chocolate, and they're so busy eating the chocolate that they loosen their grip on Juliet, aka actin. So now once the chocolate comes in, and by the way, what is chocolate representing?
The other two proteins.
Nope. Let's think about in real life, and that's why I chose chocolate. Chocolate is made up of what? Besides cocoa.
I guess milk,
Milk.
sugar.
And what do you find in milk?
Calcium.
Calcium. So the chocolate represents calcium. Okay. So when you provide calcium to the parents, troponin and tropomyosin, they loosen their control over actin. Now myosin heads can come and bind and then contract. Contraction happens.
Ah, okay, I get it. So without the calcium, myosin, and actin, they couldn't interact. Okay, that
That's
makes
right.
so much sense.
Why can't they interact?
Because
The parents have a really strong grip, right? And what's the parents? What's the parents?
myosin, right?
No.
The tropomyosin.
Yep, the tropomyosin and troponin.
Okay, yes.
Okay. All right. So that is like 50% of contraction already. So you already understood 50% of contraction by understanding that one piece. Okay. All right. So we are out of time. Watch the video. The video does cover the full steps of contraction, but now you have at least part of it. Okay. I do have a slide on there here going through the step-by-step of what happens. It's best to really watch the video because this is kind of the full step-by-step of what happens. Okay. There are some stuff at the beginning of the nervous system that you want to make sure you know, and that includes kind of the categorizing of the different categories of the nervous system. So the nervous system overall is divided into two main systems, and that would be the central nervous system that includes the brain and the spinal cord, and then the peripheral nervous system that includes the sensory and the motor nervous systems. Okay. Now the sensory, essentially that's telling you the direction in which the signals are being sent. So with sensory nervous system, which direction is it going from and to? So with the sensory nervous system, the signals are being sent, but in what direction? Where is it coming from, and where is it going to?
It's coming from our senses, it's going to our brain.
Yes. Okay. So it's coming from the environment, And that environment can either be internal or external. Like you can sense the temperature, or you can sense the calcium levels inside of your blood. Okay, so it's going to be from the environment to the central nervous system. So that's the sensory. And the motor neuron then would be the reverse. It's going to be a decision that's made, an action from the central nervous system, and it's going to send it out to an effector. What are 3 possible effectors that we have? What are 3 possible effectors? Okay, so it can either send it to a muscle, a gland, or another neuron, depending on where it needs to go. Okay, so 3 possible effectors. You can have the muscle, the gland, or a post neuron. Okay, so that's the part of what the motor system is doing. Okay, now within the sensory, so it's receiving information from the environment, whether it's external environment or internal environment, and it's going to send it to the central nervous system. But it can do so in 2 ways. Okay, one is somatic, and the other one is visceral. Okay, so what's the difference between somatic and visceral?
One refers to our movement and the other one refers to our internal organs.
Sort of, yep. So with the sensory, somatic versus visceral, oftentimes we can associate with movement, but it's not necessary movement, but it's more of it's more about the control system, right? So if it's somatic, it's something that you have more control over. And if it's visceral, and you're right, it usually is associated with internal organs. And that is oftentimes through systems that you don't have control over. Okay? So same thing. So both sensory and motor have somatic and visceral. So you can see that, what makes a lot more sense is if we look at motor. With motor, so when you're sending it out to either the muscle, the gland, or the neuron, you do it through either the visceral system, which is also known as the autonomic nervous system. So this one, you don't have control over. It just automatically sends it out. It sends it to things where you don't have control over. Where if this, yeah, go ahead.
Yeah, just quick question. Is that the same as basically voluntary and involuntary, or?
That's right.
Okay.
Yep, that's exactly it. Where somatic, so visceral is also known as the autonomic nervous system, and that's involuntary. And somatic, right here, is oftentimes through voluntarily systems. Okay, so it goes to your skeletal muscle. Okay, so same thing with the sensory. Things that are sent to the central nervous system can either come from more of your involuntary or more of your voluntary responses. Okay, and likewise, same with the motor system. So with the motor system, you have visceral and somatic, and visceral is your autonomic or your involuntary, and somatic is your voluntary. So in other words, it's going to go to your skeletal muscle, and that's what you have control over. Now, if it is visceral or otherwise autonomic nervous system, it is voluntary. And under that, there is even a further subdivision of the motor visceral nervous system is the somatic and the parasympathetic. I'm sorry, it's the sympathetic and the parasympathetic. Okay, so these are the subdivisions of the visceral. What's the difference between sympathetic and parasympathetic? They're both involuntary, but what's the difference between them?
One controls the flight or fight system, and the other one controls the parasympathetic, is the relaxation.
Yeah, your rest and digest, exactly. Okay, so for example, you can have a signal being sent to your heart. Okay, so would that be visceral So if it's a motor signal sent from your brain to your heart, is that going to travel through visceral or somatic?
Somatic.
The only time it goes to somatic is if it goes to a skeletal muscle. Is your heart made up of skeletal muscle?
No.
No, it's made up of what?
Of
So not skeletal muscle, but
muscle.
it is a muscle, but what muscle is it?
Cardiac.
You got it. Perfect. I knew you were going to get there.
I was like, ah.
I believe you. Just have to kind of think it through. And plus, when you are asked on the spot, you're like, you know.
Yeah.
But I knew you knew it.
Thank you.
So you're right. It goes to the cardiac muscle, and cardiac muscle and smooth muscle, both of them are involuntary, right? So if I were to send a signal to the heart, it would not go through the somatic, because to go to the somatic, it would be a skeletal muscle. And because it's a cardiac muscle, it will go through the visceral route. Make sense?
Yes.
Okay. So the signal is going to be sent through the motor visceral route. And if the result is that the speed of my heart rate increases, would that be so sympathetic or would it be parasympathetic?
Sympathetic.
Perfect.
Sympathetic.
Because of your heart rate increasing, right?
Yes, that's right. Because my heart rate increased, and that's a part of the flight or fight response. I'm stimulated. Okay. If my heart rate slows down, then it would be through the parasympathetic because I'm resting, and part of that is the body slowing down. Okay. Does that make sense?
Yes.
Okay. So let me give you a scenario. So, it's nighttime, it's super dark, and you're walking to your car, and you hear some noise behind you. Without even consciously doing it, your muscles tense up. Okay. What would that be an example of?
SNS.
Which one?
Sympathetic.
Perfect. Yes. Exactly. Okay. So that's an example of how it's involuntary and it's getting you ready to either fight or run away. Okay?
Could you give one more example of the parasympathetic?
Absolutely. So you know how oftentimes you have a celebration at home and you eat a lot of food? What happens when you eat a lot of food, especially like for Thanksgiving, since it's coming up soon? When you eat a lot of turkey, what happens to you?
Okay, so you have to digest it.
Yeah, and when you have to digest food, what happens to you? Are you like active or are you like lethargic?
You're lethargic.
That's right. So that's part of your parasympathetic.
Okay, because we're conserving that energy
to digest the food. So then the energy from everywhere else slows down.
Okay, thank you.
Yeah. Okay. So that's how the nervous system is broken down. And that's why in the video lectures, you will have a video lecture on the central nervous system, and then you'll have a video lecture on the peripheral nervous system. Okay. And then you will have I think you have a third. No, I think we only put it into 2 videos. So it's just those 2. Okay. All right. What else? Okay. So then from there, from the peripheral nervous system, they're just bundles of nerves, right? And if we take a look at those bundles of nerves, bundles of nerves are made up of individual neurons. Okay. So in your nervous system, you have 2 types of tissue. Okay. You have the neurons and then you have the nerve glial cells. Which one is more abundant?
The neuroglia.
Yes, because those are the supportive ones. They don't carry signals, but they're supportive. So they help the neurons thrive and do well. They protect the neurons essentially, right? Now, the neurons have the unique ability that they can carry the information. Okay. So they have conductivity. They have what else? What are some characteristics of neurons?
Excitability.
Yep. Conductivity, excitability. What else?
Secretion.
Very good. Okay. So it's excitable because it can receive signals. It's conductive because it can carry the signal from one end to the next, okay? And it can secrete at the end, okay? Great. So let's take a look at a neuron. So you can see the neuron has three main parts. What are the three main parts of a neuron?
Dendrites, axons, and synapses? Or
Close.
organizational terminus?
Close.
Cell body.
There you go. So dendrites, soma, or else the cell body, that's the same thing, and the axon. So those are the three main parts of a neuron, okay? So 1, dendrites, 2, the soma or the cell body, and 3, the axon. Now the axon consists of several parts, and that's what you listed off for me. So the axon includes the axon hillock right here, the axon length itself, the axon terminal, and the synaptic knob. Okay, so all of that A, B, C, D is part of the axon. So the neuron consists of technically three parts: the dendrites, the soma or the cell body, and the axon. Okay, so the axon is responsible for carrying that information onward. So you can see that with the structure of this neuron, can information be sent both ways, back and forth, or is it only going to be going in one direction?
From where to where, I'm sorry?
Sure. So from the structure of this neuron, okay, based on what you know about this neuron, can signals be sent both ways, like from the dendrites to the axon and the axon back to the dendrites, or can it only go in one direction?
I think it can only go in one direction.
That's exactly it. Okay, so signals can only go in one direction, which is why in the peripheral nervous system you have the sensory, which goes from the environment to the central nervous system, and the motor, which goes from the central nervous system to the environment, because neurons can only carry information in one direction. Okay, and that's why you have two within the peripheral nervous system, one for going one way and one for going the other way. It's like a highway, you have a highway going one way, a highway going the other way. They don't share. Okay, all right, so to help us understand that a little bit more, if we know about the function of those three main parts of a neuron, that will definitely solidify it. So what do we know about the dendrites? What do we know about the dendrites?
They're off the soma,
They're connected to the soma, right? What else?
and they receive and give information to the soma directly, right?
Yeah, that's key right there, right? Is that they're able to receive signals. And that's because the dendrites right here, on the dendrites, you will have receptors for neurotransmitters. Okay, so you'll have receptors for neurotransmitters. And that's the only place you will find receptors for neurotransmitters, which means that only at the dendrites can it receive the signal. It can't receive signal anywhere else. And that's why it has to go in one direction. Okay, so the reason why the dendrites can receive the signals is because they have the receptors there for neurotransmitters. So all of this communication within your nervous system occurs through chemicals or through action potential, so electricity. Okay, so through chemical or electricity. Okay, so kind of keep that in mind for a little bit. We're going to come back to that concept, okay?
But Dr. Boo, isn't that the same though? Doesn't chemical
No,
reactions produce electricity?
it does, but they're not the same though.
Okay.
Yep,
Oh,
and that's a
okay.
great question. So since you want to know, let's talk about it. Let me grab a wire. Here's a wire. Okay, so can you see this wire right here? If it's plugged
Yes.
in, if it's plugged into the outlet, okay, if I stripped it and touched the wire itself, what can happen?
You will feel the electricity.
That's right.
Because you have no conductor.
Yes, I will feel the electricity, and you would have to call 911 for me, right? So, and that's because with electricity, in order for the electricity to pass or to be sent, so you have to have something generating the electricity. It has to have a medium to travel along, and then to transfer that signal, it has to be connected to it. Do you see that? So this is one way of carrying signals, is through electricity. But with electricity, it has to be in direct contact. Okay. So let me pose you a question. You studied the neuromuscular junction. Does the synaptic knob ever come into direct contact with for example, the muscle?
No.
No.
It doesn't. And that's why it's got to go from electrical action potential to chemical, a neurotransmitter like what neurotransmitter?
ACH?
You got it. And that's why it's got to switch from electrical to chemical, because chemical can cross spaces. It doesn't require direct contact like electrical does. That's
Thank
why
you.
they're not the same. Okay?
Thank you.
Yeah. All right. So number one is dendrites, which receive the signal. Soma would be what? What's the soma? The cell body. What does it do?
It has a nucleus?
Yeah, it does. Yep. It has a nucleus. It's got mitochondria. So it's like the cell. It contains, and that's why it's called the cell body. It contains the organelles, the cytoplasm, and everything it needs. But essentially, it's going to receive the signal and pass it on. So that's the job of the soma of the cell body. Okay, is that it receives the signal and passes it on. What is it going to pass it on to?
So it's like the cell tower.
Yeah, exactly. That's a good analogy. Great job. Yep. And then it's going to pass it on. What is it going to pass it on to?
The axon hillock?
The axon hillock.
Axon.
Technically the axon, but if we wanted to be specific, we would say the axon hillock. Now, the axon hillock is really important. The axon hillock is also known as the point of no return. Okay? From the dendrites to the axon hillock, that period of time is referred to as a local potential. Okay? So local potentials can be graded, it can be high, it could be low. Okay? So it's going to be carried either way. But once it reaches the axon hillock, to pass the axon hillock, it's got to reach a certain level. Okay? We call that threshold. So the local potential between the dendrite to the axon hillock can be graded. In other words, it could be high, low. It could be, it could be detrimental. It could be detrimental. In other words, it could just completely stop if it's too low. Okay. It can be reversible. In other words, it doesn't have to be carried. Okay. So that's why we call it a local potential. But if it's high enough, if it reaches threshold, and by the way, what's the magic number for threshold?
Minus 55 millivolts.
Minus
Exactly.
55, yeah.
So you can think of it as negative 55 millivolts, or you can think of it as a difference of what? It's going from 70 to 55. That's a difference of
25.
close, 15.
Yeah.
So it's a difference of 15 millivolts. Okay. So it's got to cause it to go from negative 55 to, it's got to cause it to go from negative 70 to negative 55, or you can think of it as a 15 millivolt difference. Okay.
Does
Once
it
that
have?
happens, yeah, go ahead.
Does it have to reach threshold?
Yes. For it to convert from local potential to action potential, it must be a minimum of 15 millivolt difference.
Okay.
Yep. So let me give you an example. Okay. So here's your finger. Here's a lighter. I turn the lighter on. It's there. Well, it's sending the signal, but it's not strong enough for that signal to be sent to my finger and then up to my brain for me to know that it is hot. It's going to come closer, 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 closer. Oh, that's about where I can start to feel it.
So minimum 15.
And that reached the minimum of 15 millivolt difference. So that signal, that ability to sense that temperature is then received and then sent to my brain.
Thank you.
Yep. And that's why it's graded, right? Here's when it's receiving the signal really weakly and not enough for me to sense it. And then here's a little bit stronger, here's a little bit stronger. Oh, and that's the magic spot where I was able to start feeling it. Okay. So that's why it's graded, right? Now, once it reaches the axon hillock, that signal at this point is going to be sent to my brain. It can't stop. And that's why when you reach threshold, that kind of that area is referred to as the point of no return. It's like, you're slowly, gradually going up a roller coaster. And once you reach the peak of the roller coaster, you can't be like, stop, I want to get off. No, you're going down, my friend. Okay. So the axon hillock is the point of no return, because at that point, it doesn't matter the strength of the signal. It's just going to continuously be pushed off. At that point, we call that the action potential. And we'll talk about a little bit more about how it goes from depolarization to repolarization to depolarization, repolarization, until finally you move along to the synaptic knob. Okay, so keep that in mind. We'll talk about that when we get there. All right. But that's the neuron. So you have the dendrites, which receive the information, sends it to the soma. The soma supports the whole neuron overall, and then it's going to send that signal onward to the axon if it's strong enough. And if it's strong enough, then it's going to go and then reach the synaptic knob, at which point it's got to convert from electrical to what?
Chemical.
Why?
Because it goes through the knobs, so it goes into chemical messengers, right?
But why? Why does it have to convert to chemical? Why can't it just pass electrical onward?
Because it doesn't make direct contact.
That's it. You got it. And that's why you have to learn about how it reaches the synaptic knob, releases ACH. ACH crosses the synaptic space, binds to the receptors on the motor end plate or the junction of folds, the receptors there, and that's what then causes that signal to become regenerated again, that action potential along the muscle. And then that takes over from there. Okay? All right. So that's the neuron. Then if we were to look at the spinal cord, so make sure you know parts of the spinal cord. Now, I did notice a few of you, more than normal, were not able to distinguish between the posterior and anterior. Okay? So whenever you see the spinal cord, you will find that it folds in deeper at the posterior end. So do you see this fold right here? Okay. So it folds in deeper at the posterior end compared to the anterior end. And that's kind of one giveaway as to what's the front and what's the back. Okay. Because a lot of you wrote down, for example, posterior horn when it was the anterior horn on the lab quiz. Okay. So I just want to make sure you can tell the difference between posterior and anterior on the spinal cord. All right. That's the spinal cord. Make sure you know how, and it's really cool actually. Once you learn this, it's like, oh, it makes sense. You don't have to memorize it. Okay. And one of the cool things about this is, some students get really tricked over between the posterior root and then posterior root ganglion. You shouldn't. It makes a lot of sense once you place where the neurons fit in. Okay. So let's do that really quick. Okay. So here's the spinal cord, and I have two neurons in there. I have this neuron right here. And as I'm tracing it, can you tell me what kind of neuron this is?
It's a motor neuron, right?
I love it. Thank you. It's a motor neuron, and you are right. Okay. So if you look at the size, what is larger, the soma or the synaptic knob?
The knob.
I thought it was the soma.
I would agree with you. It's the soma. Yep. And that's why you'll notice that the anterior horns are larger. They bulk up a little bit compared to the posterior horns. So the anterior horns tend to be a little bit more chunky. Why? It contains all of the somas of the motor neuron. Okay. And not only that, if you look at the anterior root, there's no ganglion in there. It's just the anterior root. There's no anterior root ganglion. And that's because the soma is already found in the anterior horns. Okay. Let's move on to the next one. Okay, here's another neuron. Which neuron is that?
Sensory.
Yeah. Which means most likely there's another neuron connected to that going out like this. Probably like in your hand so you can feel touch, pressure, so on. Okay. So with a sensory neuron, it's going to receive information in the environment. It's going to pass it onward. Okay. But if it has to go really far, oftentimes it's got to rely on a second sensory neuron. And if that's the case, that soma has got to be on the outside. And that's why here, you see how it gets bulky right here? Here. It kind of swells up a little bit. And the reason for that is because it has to house the soma of a sensory neuron. And then if you look here at the posterior horns, it tends to be a little bit smaller. Why? Because it only has to house the synaptic knob of the sensory neuron. Oh, that's why certain areas are thinner than others. That's why the posterior root has a posterior root ganglion. Ganglion means cell body, by the way. Okay, so whenever you hear the word ganglion, it means like an area with cell bodies. Okay, or like a cyst, a mass. Okay, so that's why the posterior root has the additional structure of the posterior root ganglion, where the anterior root doesn't. And that's why the anterior horns are a little bit more chunky than the posterior horns. Okay.
So
Question.
the posterior root ganglion carries the posterior root and the somas from the sensory neurons. Is that?
Carries just the somas of the sensory neurons. It connects to the posterior root, but it doesn't carry it. It's just two different areas.
Okay.
Yeah. Okay. All right. All right. Then the spinal cord has 31. You need to know that there's 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccyx. Okay. The way I remember the first 3 at least is you have breakfast at 8, lunch at noon, and dinner at 5. Okay. Anything you use to help you remember is an advantage. Okay. So keep thinking things through, and then I'm like, oh, this helps me remember, then use it. Okay. Then with the brain, make sure you know parts of the brain. A lot of you did amazing jobs on the brain. So we'll leave it there. So you went through the parts, the 4 main parts, the cerebrum, cerebellum, diencephalon, and brainstem. And then you did the subregions of them, which include the, if you're looking at the cerebrum, you're looking at the lobes. If you're looking at the diencephalon, it's divided further into the thalamus and the hypothalamus. And then if you're doing the brainstem, you've got the midbrain, the pons, and the medulla oblongata. Okay? But I want to save my time for the most difficult, or what students tell me is the most difficult. And that's this one. Okay? All right. So we're looking at potential. That means how electricity is sent through the neuron. You have resting membrane potential, local membrane potential, threshold potential, and action potential. Okay? So let's really do a deep dive into this. Okay. Anytime you have resting membrane potential, it is referred to as polarized. The term polarized, by the way, means negative. Which is why at this point the membrane is negatively charged. Okay. So let's just draw it out. Let's draw a quick neuron. Okay. We'll do the dendrites just to make it complete. Okay, so it turns out that along the axon, there are these gates and channels. And in particular, there's some for sodium, there's some for potassium. Note that I put positives on both, okay? I put positive signs on both. So they're both what type of ion?
Cation.
Perfect. Thank you. Okay. So on the inside, okay, at resting membrane potential, you're going to find potassium, lots of potassium inside. Okay. And you're like, wait a minute, potassium is a cation. So isn't that what charge?
Negative.
Negative.
No. Isn't that what charge?
Positive?
Yes.
Positive.
You see the plus and the K? That means it's positive. So what's going on here?
Because the inside of the cell has other proteins that are negatively charged and take over
You got it.
the other one.
Mm.
That's it. That's what I'm looking for. So on the inside, let's do purple. I have negatively large negatively charged proteins that don't leave, that can't move through these channels real easily. Okay. So even though I have potassium on the inside, which is positively charged, I have large things that are negatively charged. And that's why at resting membrane potential, if I measure that, it's still overall negative 70 millivolts. Okay? That nothing's happening at this point. This is just how it normally is. Now on the outside, okay, in the extracellular fluid, there's always going to be lots of sodium. Okay. All right. So let's say I send my brain right here. This is a brain. The brain sends a signal. And that signal is going to be through the form of a neurotransmitter. So here are the neurotransmitters. And here's my neuron. Here's my dendrites. Okay. The neurotransmitters bind to receptors in the dendrites. That's where they receive the information. And now a smaller potential depolarization occurs along here. Okay. For that potential, for that depolarization to occur, there's also these channels here that open. And when they open, automatically what happens?
The sodium comes in.
That's right. Sodium comes in because it's abundant on the outside, right? So small amounts of sodium come in, and that's what causes the membrane to change. But it has to be enough change so that when it reaches this point right here, it totals a what net change?
Action potential.
It has to reach threshold, right? Which is a
Negative 50
millivolt. Exactly. So local membrane potential is where it receives the stimulus and then it sends it along from the dendrites to the soma. And if it's strong enough, okay, it's going to reach threshold.
Local potential is where it receives what did you say exactly?
Signals from the brain. Okay. So it's going to be where it receives signals from the brain or actually if this is a motor neuron, it will receive signals from the brain. If it is a sensory neuron, it will receive information from the external environment. Okay. So it depends on what it is, what direction it is, what kind of neuron it is. Is it motor or is it a sensory? Right now, I'm giving an example of a motor neuron. Okay. So that's a local membrane potential. So from negative 70, it's going to kind of go up, and in other words, become less negatively charged because now it's receiving small amounts of sodium, and sodium is positive, right? It's just imagine this way. You have a debt. Okay? And if you start working, each day you work, you start to be able to pay off a little bit of that debt. So you become less negative. And that's why it goes from negative 70 to, let's say, negative 69 to negative 60 to negative 59. But it's not until it can reach negative 55, a 15 millivolt difference, that then it can switch from local membrane potential to an action potential. Does that make sense?
Yes.
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 Perfect. Okay. So what can you tell me? Summarize what I just told you. Local membrane potential occurs where?
Dendrites. The
Yep.
soma.
From dendrites to soma. And you can even say kind of like the axon hillock. Okay. Some characteristics of the local potential.
Reversible.
Yes.
Decremental.
Perfect. Okay. So it's graded as well. So it can be in various strengths. Okay. So threshold then is when that signal is able to achieve a 15 millivolt difference. In other words, it's able to go from negative 70 to what?
Negative 55
At what location?
at the threshold.
The axon hillock.
There you go.
Oh, okay.
Yeah. I mean, you were right, but one is the actual location where the other one is the type of potential. Okay. All right. So achieves 15 millivolt difference. In other words, it goes from negative 70 to negative 55, and that's when you reach an action potential. Okay. Now, in an action potential, strat, at this point, are we concerned about the dendrites anymore?
No.
No, not at all. Oops, I do that a little bit too big. I'll go like this. At this point, I'm concerned about this portion right here. Okay. All right. So we're going to use some imagination. Oops, sorry. Okay. So imagine that you have a ball on water, a plastic ball on water. Now, if that water is stationary, will that ball move at all?
No.
It won't. Exactly. So a ball on stationary water isn't going to move. So just like this, okay, that signal that now is on the axon isn't going to move unless there's some waves, some changes within the axon. And that's why that area, in order to move that signal from one location to the next, to the next, to the next, you have to create waves. You have to create changes, okay? And that change occurs through change in membrane potential. So let's say here's the signal right here. Oops. Here's the signal right now. Here's the signal, okay? This area, this membrane right here, has to undergo changes, and that's why this area of the membrane allows sodium to move in. And then when the sodium rushes in, it causes the membrane to become what?
Depolarized.
Yes, exactly. It causes it to become positive. And immediately following that, the potassium gates are going to open, which causes the potassium then to do what?
Repolarized.
To leave, and that's what causes repolarization, which is negative. So that change in membrane, depolarization, repolarization, causes kind of a wave, and that's what pushes the signal onto the next area. So here's the signal now, and the same thing happens. That it's going to happen. So in this area now, this area is going to undergo depolarization, repolarization, and that causes that signal to move further to the next area. And then you have a depolarization, repolarization. That continues to move it, and it continues to happen until it finally reaches the synaptic knob. Okay? So in an action potential, you have depolarization and repolarization consistently throughout the length of the axon until finally that signal is carried or moved to the end.
Do the nodes of Ranvier, do they have anything to do with this moving along type?
Yes. The node of Ranvier is the only areas where depolarization and repolarization does not occur.
So that's would be considered like the peak, right?
No, so think of it this way. Okay, you're an ice skater, so you're on ice, right? In order to move along ice, what do you have to do first?
Move your feet.
You have to push yourself off. You have to create that momentum. So here's the ice skater. Here's the ice skater. Beautifully, they have to push themselves off in this area. When they reach the node of Ranvier, they can just glide. And then they're going to eventually come to a stop, right? So then they have to push themselves again off. Okay? And then when they reach the node of Ranvier, where the area is covered, so you can't have a movement of the ions, they're going to glide again. And then here, they're going to push themselves off again. And then they're going to glide. And then they're going to push themselves here. And then they're going to glide. That's the node of Ranvier. The node of Ranvier allows the signal to move faster because at that point, it can just glide through. Does that make sense?
Yes.
Yes.
It's kind of cool, right? So
Yes.
no, it really is. Yeah. Like your body is like an amplification of what you see in nature, right? These are all concepts in nature, right? The ability to glide through, the ability to build up momentum, and so on. So it's very cool. Okay. So that's what happens in an action potential. So there's areas where there's no myelin. Okay. Oh, sorry, I switched them. So there's areas where there's no myelin, which is then the node of Ranvier's, and that's where you're pushing off. And when there's an area where there's myelin, it's covered, that's where you glide. Okay?
So, question.
Yeah.
When they have the myelin and it has to go through inside the cell to keep on
Yes.
going, it loses electrical charge because everybody's pulling from it. My understanding of the node around there is that when it reached the node around there, it basically gave it a charge again. So it pushed it even further.
That's exactly it. Yep.
Okay.
And that's what I corrected myself because I had switched it before. So now I'm correcting. Wait, okay. So the node around here is where it's not myelinated. So wherever it's not myelinated, that's where it has to push itself off.
Okay.
Yeah. And where it's myelinated, it's covered, it can just glide through. Yep.
So is that how it allows like the potassium to flow out of the cell?
Yeah. So where there's node of Ranvier, there's no myelin. And that's the areas where you can have the sodium enter and potassium leave.
And in the sorry, go ahead.
No, I was finished. Go ahead.
So in the myelinated areas, does the signal actually speed up?
Yes.
Okay.
Yep. Yep. And that's the definition of myelin, right? It insulates and speeds up electrical impulses because you don't have to waste the time of pushing up, depolarization, repolarization, depolarization. It's just gliding.
Right. Okay.
Yep. Yep. Now, what happens though, once action potential is done, is this neuron the same exact as the previous neuron? Is it in the same state as it was before?
No.
No, because before on the inside it's potassium. Afterwards, now what's inside?
Calcium.
Sodium.
Sodium.
Right? So once an action potential has been pushed through, it actually has to go back to being stable. In other words, it's got to push the sodium back out and push the potassium back in. And that's where we call hyperpolarization. Okay, because too much, because potassium tends to leave at a faster rate than sodium can go in. So it's got to kind of reverse the cations and then go back to where it was at -70. Okay. But that's essentially how signals are sent. Not too bad, right?
So question, the hyperpolarization is not when you have way more potassium inside than what you needed? No?
Hyperpolarization is when it becomes too negative. And the reason why it gets too negative is because potassium leaves at a faster rate than sodium can go in.
Okay. So it drops more lower than -70, and then
That's right.
and then it moves it's a little bit higher to continue on resting.
Yep, it has to reestablish itself. It's got to switch the cations, and then it's got to go back from -75 back to -70.
Okay.
Yep, and that's what you'll see here. You see this image right here? So I have A, B, C, D, E, F right there. F is where it's hyperpolarized, and that's because too much potassium left.
Too much potassium?
Yep, too much potassium left, and that's why it goes from -70 to -73. I'm sorry, yeah, to -73 or 75. It just gets more negative.
Thank you.
Is that when the pumps close? Is that what where the pumps start basically working to restabilize the resting potential?
Exactly.
Okay.
You got it. It's got to switch it back over, and that requires energy. Yep. Okay. All right. So that essentially is it.